I am a neuroscientist, so I'm interested in how the brain controls complex vocal behavior in health, in disease, and also in age. But today I'm going to be taking a little bit of a different direction and explain to you some of the findings that I found in the vocal fold muscle, looking at gene expression changes that may coincide with vocal and swallowing changes in our translational rat model of Parkinson's disease. Parkinson's disease is a progressive degenerative disorder. It affects approximately 10 million people worldwide, and that prevalence is really expected to increase as the population ages, which is going to significantly impact our healthcare system. The hallmark pathology of Parkinson's is dopamine loss in the substantia nigra, as well as the accumulation of different types of proteins in various brain regions. And together, these really lead to the classical signs that you're probably familiar with, which are bradykinesia, instability, tremor, and rigidity. But Parkinson's is really has a whole host of other comorbidities that go along with it, including things like cognitive deficits, anxiety, and depression. And what I'm interested in dysphagia, but also dysarthria, which is disordered voice. So voice issues in Parkinson's really impact approximately 90% of people with a diagnosis. They are refractory to standard pharmacological therapies, and they really decrease quality of life in individuals with Parkinson's. And this is due a lot to the fact that we really don't understand what the mechanisms are underlying these early onset vocal changes that significantly impact the quality of life. We really don't have an understanding of what is happening in peripheral structures as well, because we know now that Parkinson's is really a whole body disease. It's not just a central nervous system issue. It is nearly impossible to study early onset Parkinson's in humans due to environmental variability, methodological control, and also the inability to obtain early pathological tissue. Therefore, we model early progressive Parkinson's using a genetic rat model called the Pink1 knockout rat. So my work over the last 7 years on this rat model has really shown that these rats are very much analogous to what you would find in humans. So they have the appearance of vocal motor deficits that appear early on in life, and they progressively worsen. The timeline is in the center there. Worsen with age, so about 8 months of age is where I like to kind of focus my research. These vocal deficits are very similar to what you would find in humans, progressive in nature. They significantly impact the ability to communicate with the conspecific. So female rats really don't approach a male rat with these types of deficits. They are non-responsive to levodopa, so you can give them the same type of dopamine replacement that is supposed to alleviate some of the classical hallmark pathologies, and it doesn't do anything for vocal behavior in this model, similar to what you would find in a human. We do know that this rat model also exhibits early oral motor function, so changes to chewing and swallowing. They have the onset of limb motor deficits about 6 months of age, and these behavioral changes really correspond nicely to what you would find pathological changes in the brainstem, so decreases in neurotransmitters, increases in alpha-synuclein aggregation, and then also changes in the peripheral structures. So we've started to examine the vocal fold muscle, and we see changes, alpha-synuclein aggregation in the periphery, as well as changes to muscle fiber type. So the central hypothesis for this work is that loss of PINK1 within the thyroarytenoid muscle, which is the muscle that controls the vocal folds in both the humans and the rat, really alters genes and biological pathways that are important for appropriate vocal and also swallow function. In this study, we took PINK1 knockout males and compared them to age-matched wild-type controls at that 8-month time point where we have a really good idea of what their behavior is like and also some of that early onset pathology. We took RNA from the left and right thyroarytenoid muscles. We used an Illumina HiSeq 2000 platform to run high-throughput RNA sequencing, and we mapped it to the rat genome. And we did 3 kind of main statistical approaches. So one is looking at differential gene expression. So what genes are upregulated and downregulated between genotypes. And what you see here is that there were 157 annotated differentially expressed genes. Only 4% of those were muscle-related in nature. A lot of them were metabolic, cell signaling, genes, and some of them were epidermal in nature. Actually, a lot of them were epidermal in nature, and if you have any insight to that, I'd love to talk to you at poster number 2 later. We then did a weighted gene correlation network analysis, and we followed that up with gene enrichment tools using Enricher for overlap with human databases and other disorders. And what you just see displayed here then is the module in which PINK1 was the most significant gene, and then mapped to PINK1 are this network of genes. So genes that are corresponding and working with PINK1 in nature. We ran that specific module of 75 genes through enrichment software, and what we did was we tried to categorize molecular pathways that may be modulating voice and swallow, and some of those interesting pathways have to do with proteasome degradation and also different signaling pathways. Several genes suggest overlap between multiple disorders, including that of Alzheimer's, ALS, and dystonia. There were 4 genes that really stood out to me. 2 of them that were significantly upregulated were for inflammation and apoptosis. 2 genes that were significantly downregulated had to do with alpha-synuclein cleavage, which we know that alpha-synuclein aggregates in the vocal fold muscle, also the oxidative stress and neuronal protection. Our data correlates significantly to human idiopathic gene sets, as well as the PARC2 and PARC6 data sets, which really lends to the validity of this model. And genes identified here may be used as biomarkers for treatment development for voice and swallow deficits, potentially across disorders. And I do have new funding that's going to address the hypothesis that we're going to use these data along with drug repurposing tools to then hopefully identify and validate treatments. So really combining behavioral data, genomic data, and ultimately pharmacological manipulation. Thank you.